Our next speaker not only has a wealth of information, but he's also kind of become a celebrity in this industry. Dr. Juan Osorno is a veteran professor in the Department of Plant Sciences at North Dakota State University. He's been there since 2007. Dr. Osorno is a prolific dry bean breeder / geneticist, having developed and released over 20 cultivars and improved germplasm lines throughout his career. Beyond the breeding laboratory, Juan shares his knowledge by teaching general genetics to about 250 undergraduates every year and leads the graduate seminars. He's a bit of a road warrior, often speaking in the scientific community, making over 40 domestic and 25 international presentations on 5 continents. His experience and expertise extends beyond NDSU. He also serves as a technical scientific advisor to breeding programs in Guatemala, China, Zambia, Colombia, and others. Please welcome Dr. Osorno to Bean Day 2025.
Let's see. Can everybody hear me well? Yes? I'm gonna move this because I'm gonna stumble into it. Yeah. One way I release energy when I'm presenting is I move a lot, right? So, yeah. Thanks for the opportunity to be here and showcase all the research that we do in our breeding program. It takes a village. I mean, I have the privilege of coming and telling you all these things, but it's a group effort. There is a lot of other people, many people involved into all this, not just even within NDSU, but we have a lot of collaborations both with other universities and private companies as well, right? Okay, so before I start, this is a bit of an announcement. Remember to collect your dry bean variety trial bulletin. Has all the information about the trials that we did, what we do every year, right? My team coordinates 4 of those trials in the region, 2 in Minnesota, 2 in North Dakota, plus you have all the research and extension centers also adding their information here. Dr. Ana Carcedo is the person now in charge of compiling all these reports. So it's also available online. If you just Google NDSU variety trials and it's gonna pop. And that's the link also. And then you can get an electronic copy if you don't get one of those. If you still want the paper copy because that's what you like to read, just reach out to me and I'm happy to send you some via mail, okay? No problem. All right, what are we gonna talk about today? We have several things to cover as always, and not enough time, right? I only have half an hour to talk to you. We have a new black bean variety I'm gonna be telling you about. Also, as usual, I like to say a little bit words about how our varieties are doing across those trials that were shown here. A little bit of what we're doing regarding disease resistance, and a really interesting story about Pinto seed coats and some of the things that we're doing there, some of the findings. Finally, a little bit about what we're doing regarding white mold, probably our #1 disease, as we all know. So, yeah, let's get into topic, right? I present you ND Galaxy Black Bean. Pretty cool name, right? Hopefully you like it. So it follows the tradition of our star-related names or space-related names, right? We had Eclipse, we got Twilight, and now you have Galaxy. We're pretty excited with it. It's across all the trials that we have been testing this variety, it yields about 100 pounds more per acre than our regular chicks, right? Most of them, with the exception of 1, okay? And I'll show you that data later. 100 pounds per acre, yeah, you might say, that's cool, but that's not a lot, but that's the way things happen in beans, right? Small increments, as always. 100 pounds per acre also means 30 to 40 extra bucks per acre in your farm operation, right? So I think that's significant. We're also excited because among blacks tend to be very susceptible to common bacterial diseases. And this one, we were able to, because of the parents that we were using in the pedigree and the development of this, we got a really good parent with high levels of resistance, and luckily that resistance kind of came through as well. Really good plant architecture, canning quality, very similar to what we're used to see in in other varieties. But here it is, right? Everybody cares about yields. Don't tell me all that stuff, Juan. Just tell me how much it yields, right? So you're looking at data collected across 20 to 30 environments. I think in the case of the comparison that you see between Galaxy and Blacktails, which they were pretty similar, right? But they were tested in less locations than the others. The others were across 30 locations. Blacktails is kind of a newer kid on the block coming out of the Provida program. The Schallenberger brothers are here also, my colleagues. Pretty good, right? Two good varieties that you have there, two good options, yielding pretty much the same, 21 bags. And then you look back and see the progress that we're making. Again, these little baby steps that we're doing, increasing yield across time, right? And that's also important. And that's really important because, as Machinery Pete was saying earlier, sometimes we need perspective. Sometimes we look at 1 year data and we're like, we're not doing anything, right? But remember, we're producing today twice as much in that same acre of land that we were producing 50 or 100 years ago. And that's what that graph in your left is showing you, right? Look at the increment in the last 100 years in terms of yield. We duplicated yields pretty much in that amount of time. And there's a lot of studies that show that a lot of those increments are caused by just better genetics, right? We have talked about that before. I actually had an intern last year in my program, Sarah Long, and I asked her, why don't we look at what has been the yield gains in our breeding program? Forget about everybody else, forget about, kind of a selfish experiment, right? Let's do an assessment of if we are really increasing yield in our breeding program or not. So she looked at 70,000 data points that she was able to collect, right? And compile all these. And then in that table, you see that, for example, for blacks, in the last 35 years, we've been increasing 5.8 pounds per acre per year. That's our yield gain. Kidneys, 13.8 pounds per acre per year, right? Navies, probably the lowest one. Navies are tough, man. Navies are tough. And pintos, about 8 pounds per acre per year. So positive gains in 35 years. That's why, again, I think that was very savvy from Machinery Pete saying, look at the longer term rather than just looking at 1 year of what's going on, because it gives you a lot of perspective, right? We good? When it comes down to other agronomic traits that are important for us in the industry, and in terms of agronomy as well, I think we're on par with all the other commonly grown black beans in the region. When it comes to seed size, and we use 100 seed weight as a proxy to measure that seed size, right? So the higher the number, the bigger your seed, pretty much. And we're there on par with everything else. Days to maturity, 97 days. When you have eclipse at 96, 1 day of difference really doesn't mean anything. We know that. At least doesn't have any biological meaning. Plant height, among the highest, right? Days to flowering is fine, but I want to call your attention to this. When we were screening, both in the greenhouse and in the field, for common bacterial blight diseases, we use a score of 1 to 9, where 1 is really resistant, almost no symptoms, and 9 is really bad, right? So it's just a visual scale that we use. So 1 to 3, we consider it resistant, 4 to 6 will be intermediate, 7 to 9 is susceptible. And look at that. We got a 2.8 across all the experiments that we did for Galaxy versus 4s and 5s for everything else. So there is an increase in the levels of tolerance or resistance that you are going to have there. I'm not going to go and claim that this variety is immune to bacterial diseases, right? Because then someone is going to come and tell me, Juan, you were wrong. Look at my field, right? But it's going to do much better. And probably is not going to have any economical damage. Even if you have the bacteria there, it's not going to hurt your yields, which is really what matters at the end of the day, right? Okay, what happens with the rest of the varieties that we have been released across the years? You have 4 charts there. Let's focus first on the Pinto, upper left on your screens. And again, this is this is a very rough exercise. We're just taking the 2024 data across all the trials and locations that we have and kind of put it there. A very strict statistician will say you cannot do this because you have varieties that were grown in like 8 locations and then some others were grown in 2. This is not a fair comparison. We call this an unbalanced set of data. But there is a lot of statistical tools now and powerful software that allows you to kind of balance some of these things now. In any way, you can see that in terms of conventional pintos, the winners there were Monterey, Cowboy, and Charro. But be careful again, Charro, only 2 locations, data from 2 locations, right? So go back to long-term trends. 2 locations is probably not much of a good source of information compared with 8 for the other ones. So our variety doesn't just being average, I would say at this point. And that makes sense because we know that Falcon strives only when you have disease conditions like rust, or you have soybean cyst nematode in your fields. That's when Falcon is really gonna show up. So I guess I'm not surprised to see that. Kudos to the Provida breeding program because that's their genetics showing up there, along with Charro, which is a new variety from Michigan State University. So worth looking at if you wanna test something else. Slow dark pintos. You can see there that the champion so far is our ND Rodeo that we just released a couple of years ago. So we're pretty excited about it. There is a lot of hype about it. All I hear from our customers and people is, we want rodeo, we want rodeo. So we're pretty excited about it and see how it goes. Blacks, you can see our newer Galaxy showing pretty good there, right? So that line that you have in the middle is the average of all those trials. And then anything above the bar means that you are yielding higher than the mean, and anything below it means you're yielding less than the mean of those trials, right? And then on Navy, our Polar is also looking pretty good in the trials that we look at it. Another important thing that I wanted to point out, I always hear people saying, oh, slow darkenings don't yield the same as regular darkenings. On average, you can see there, side by side, this is the first year that we were able to do, some of you were in our trials visiting. This is the first year we were able to do a separate slow darkening pinto trial and then another conventional or regular darkening pinto trial, side by side in our locations. And look at the yields, 19 bags. They are not different at the yield per se. Maybe some other things happen later when you're cleaning your beans and you probably get more splits and cracks and that reduces your total yield. But at the field level, these things are yielding the same, okay? So don't be misled into thinking that they don't yield the same. Oh, I had some highlights there and I forgot to include them. Okay, kidneys. Sadder story, we only have 2 locations in Minnesota where we do the kidney trials. One of them failed. For the first time in 20 years that I've been doing this, we had deer eating my beans. So we have a nice population at Cordell's farm that loves eating my beans. So we're gonna have to figure out something about it. So only 1 location. Be careful, take this data with a grain of salt, right? Because it's only based on that location, and that location had flooding issues at the beginning of the cycle at Staples. So, I mean, I'm not gonna probably say much about it. Look at Great Northern. Pegasus, our Great Northern, still killing it. For me, that's the highest yielding variety I have out of any other we have in our program. I mean, I'm crossing the hell out of Pegasus, trying to move those good yield genes into other market classes and other varieties. So hopefully we can do that later. Rosalind was released last year as the new pink. And as you can see, it's just killing it compared to the usual suspects that we have for pinks. We don't have a lot of pink varieties anyway, so it's good to have an option for the farmers there, right? And Jose was the person helping me, my assistant, with gathering all this data that we collect across locations. Let's see. we have seed available for pretty much all these varieties, right? All you have to do is just contact me, contact Foundation Seed Stocks, contact your seed dealers. Many of them already have certified seed of these varieties. We have a booth on the room over there from our foundation, our research foundation, and they have information, booklets, pamphlets that you can take home and with the information about all these varieties, right? This is all for you. We're a public program. Our role is to serve you and providing all these good varieties. So take advantage of it, right? This is almost free research, right? That is happening in the way that benefits the whole industry. You have some other pictures there, some other varieties that we have released in the last few years. All right, let's talk about disease resistance. And I guess we're in a unique place. So as many of you know, we do a lot of seed production and seed multiplication out west in Washington and Idaho. The reason is because their conditions are almost disease-free, with few exceptions. It's all irrigated, it's dry, really optimal conditions. You get really high yields and really good quality. That's not the case in North Dakota, right? We get a lot of humidity, a lot of rains. We have a lot of dryland agriculture. We're more exposed to the elements and the factors that are out there, including diseases, bacterial diseases, fungal diseases, et cetera, et cetera. So we do a very conscious job trying to use genetic tools to improve. And as a proof of that, I just told you about Galaxy and the common bacterial blight resistance. But then my assistant, Dr. Nusrat Khan, he's been compiling and doing an inventory of what we have currently in our breeding pipeline with different types of resistance, right? And that's what you see there. When we talk about, in the breeding pipeline, we talk about advanced and preliminary breeding lines. The advanced are the ones that are getting closer to the release, and the preliminary are still under testing, right? And you can see that we have interesting results for resistance to anthracnose, rust, and even white mold across different market classes. So and we're incorporating that all the time, including CBB. I mean, the data is not here, but us as well, right? Okay, let's change topics a little bit and talk about slow darkening pintos again, because it's something that everybody asks me about all the time. All the time. This is a common conversation, right? If you don't know, never heard about slow darkening pintos, just Google it. You're going to find a lot of information, right? But this is a new subclass of pinto beans that allows or delays the post-harvest darkening that we normally see in our pinto beans over time, right? So now we're talking about regular darkening pintos or conventional pintos, and then we have the subclass of slow darkening pintos. You go to some places, and that's what consumer wants. You go to Mexico, that's what they want. You go to Dominican Republic, that's what they want. A lot of the dry packaging industry, that's what they want, right? I have shown evidence before in previous years that these are faster cooking. I had commercial kitchens calling me and say, your beans are getting mushy. It's like, yeah, because you don't need to keep them on the stove for so long. This is an opportunity to save time and energy, right? And we're not using it. Also research that shows that they are much better, more nutritious for you. And now we're again, proof of concept that now we are getting at the same level of performance in the field as we have with the regular darkening, which was not the case several years ago. So why? Why we have differences? Why some of these pintos don't darken and some others do? And a lot of the research that we have done in collaboration with people who are much better at biochemistry and chemistry than me, we have a really good collaboration with Raymond Glann at Cornell University. It's all about the flavonoid components that are in the seed coat. And those flavonoid compounds are the ones that are responsible for the colors that you see on the beans. There are about 4,000 flavonoids that we are aware of in plants. This is only in plants. We don't produce flavonoids, by the way. We don't really know what they do in the plant. What's their role? Why a plant produce flavonoids? There must be a reason We just don't know exactly. Besides the color, we don't know exactly. Some people argue that they might be a defense mechanism for diseases or insects or herbivores, et cetera, et cetera, et cetera. We don't know. But they are there. And this is pretty much like an atlas of different flavonoid profiles depending on the market class that you're looking at, and you can see the list of those. Obviously, when you look at a white bean, like a navy or a gray northern, you pretty much don't have flavonoids, as you can see on the left side, right? You only get to see flavonoids accumulated in that seed coat when you start seeing the color. See it? Okay. I mean, we could have an entire 30-minute talk just talking about flavonoids, but I'm not going to bore you about that, right? So I've said this many times, and I'll keep saying it more until people really get it. This brings a really interesting opportunity from a nutritional standpoint. Slow darkening pintos have more bioavailable iron than regular pintos. This is a great opportunity for nutrition, for the food industry, okay? In the left side, you have a graph showing that when you measure iron content in the seed, in between regular pintos and slow darkening pintos, they're about the same. Nothing happens. But then when you go and measure how much of that iron is actually in the right molecular form to be available for your gut to absorb it and use it, the slow darkening pintos are 4 to 7 times higher than that, okay? We have to take advantage of that. We have to take advantage of that. That's really important. And it can be all explained because your regular pintos tend to have 2 to 4 times more proanthocyanidins in those seed coats versus the non-darkening ones, the slow darkening ones, sorry. Are we good until there? So you have a bean that is good looking, does well in the field, cooks faster, and is more nutritious. I don't know what else you want, right? It's like the list just goes on. But in plant breeding, and I have several colleagues here that are plant breeders as well. I can see Ken Kamisi was over here as well. You know, plant breeding is a trade-off game. When you breed for something, sometimes you end up causing another effect into something else, right? Because a variety is a blend of a lot of good traits and good attributes that we are always wanting, right? And it's that blending, that's what makes breeding complex. If I could just focus on one trait, I could make a lot of progress, but it's just the blending. And every time you want another trait, it's like adding another layer of complexity to the whole process, like an onion, right? You add more layers, it gets more and more complicated, right? In the case of slow darkenings, we ended up seeing now, there is a lot of anecdotal evidence that shows that there is higher seed damage in the slow darkening pintos, right? That's what I hear from a lot of people in the industry across different parts of the value chain. We know that seed moisture plays a big role into damage for the seed as well, so that's a big factor. Everybody knows that and is aware of that. Harvesting beans too dry is just gonna give you more damage. That's the nature of the beast. We also have seen some issues in slow darkenings with germination and emergence. They tend to be more finicky and also a little bit more of susceptibility to root rot diseases. So going back to the hypothesis, maybe those flavonoid compounds are important for germination and diseases and things like that, right? We don't know. Anyway, that got me thinking, right? And this is really important. We tend to forget this. When we're dealing with beans, not as seeds, but mostly as commodity, we tend to forget that this is a living organism that we're dealing with. It breathes. It's alive. It's a seed, right? It's not just a bean, but it's a living organism. It's breathing. There's some biological processes going on there across time, right? That's a slide I found that they show in preschool. So they tell you this stuff in preschool, right? But we just tend to take it for granted. So this got me thinking, okay, we have the seed damage. We know that the flavonoids are into the seed coat. What if we look at what's happening in those seed coats And maybe that's where the answer, we can find some answers regarding this higher level of damage that we're seeing on pintos, right? So that's an electron microscopy picture that you see on the right, showing that any seed coat is actually, we think about the seed coat, but it's actually this multi-layer coat that has all these different sections that we call the palisade cells, the subepidermal, and the parenchyma, okay? So let's dive into this and try to understand better what's going on in this area and see if we can find some answers for these questions, right? So the first thing is my master's student, Eduardo, was in charge of this work. And the first thing he did, okay, let's try to answer that question. Is it true that we have more damage, more splits and cracks in slow darks versus regular? And that graph pretty much answered that question, telling you yes, especially for split seeds. That's your blue bar. So on the left side, you have your regular darkening pinto, and on the right side, you have your slow darkening pinto. The blue bars are split. The red bars are crack seeds. When it comes down to crack seeds, we don't see major differences, right? A little bit more on the slow darks, but the splits is really an issue. And that's what many of you have told me through conversations that I have with all of you. So, this was more of a conscious way to measure this using samples coming from the same trials. And we have done this across 2 years. So, this is pretty solid data, I would consider, that answers that basic question. Yes, it's obvious that we have this issue here. So, with the same data that he was collecting, we did a statistical procedure known as principal component analysis that pretty much takes your data and try to see common trends and common factors and start creating these clusters or groups of things, right? So you have the varieties there, and pretty much what it's showing is that it creates kind of 3 clusters or 3 groups. There is a group in red there that shows a low number. Those are varieties that show a low number of split seeds, high amount of crack seeds. A second group that is kind of intermediate is that in the middle, the green one. And a group 3 with a high number of splits but low amount of crack seeds. The most interesting part is that the square figures are the regular darkening pintos and the circles are the slow darkening. And what you can see there is that they're not grouping together. So now that starts telling you, it doesn't look like this is really caused by the slow darkening per se, right? Because you have varieties that are slow darkening that are as good as the regular darkening ones. It's a mixture, right, of different things, right? So again, that we're scratching our head trying to figure out, okay, what else can we do? So we have a really nice electron microscopy facility at the USDA in Fargo, and we talked to them to start making these sections, seed sections of the seed, right? And then we were taking pictures of different varieties for those seed codes, and not just one, but multiple images of those. And let's try to measure the thickness of these different layers that I was telling you about before, right? So that's pretty much the work that Eduardo was doing. And when you group everything by either slow darkening versus regular darkening, you see that there's actually higher variation for seed coat thickness, the total seed coat thickness for the regular darkenings, and there is less variation for the slow darkening. And they're not really that different, right? When it comes to that measurement, they're pretty similar there. And even more interesting, when you look at varieties, at each varieties, and look at that seed coat thickness across all those layers, you have on the left side, on the red, you have the slow darkenings, and then on the right side is the blue with the regular darkenings. Really no major differences, right? They all look kind of similar. You have good ones and bad ones within each one of those groups, right? We're still scratching our head. What's going on here, right? Interesting. Well, let's look at each one of those layers by separate, right? We're diving in. We're getting into this rabbit hole even more now. So the first layer, the palisade layer, you look at the data, more variation in the slow darkenings, but again, no clear pattern of, one thing is different than the other as a group, right? We just see, good varieties and bad varieties within each group. You go to the next layer, same thing. We don't find much variation there, right? Here, on the third layer, something that was really interesting was to see that there is much more variation in the case of the slow darkening regarding the thickness of that layer versus the regular darkenings. But again, you see a mixture of different things where within each group you have good and bad varieties, right? So now we go ahead and say, okay, how this correlates with the damage that we were just talking about. So you go ahead and do another statistical procedure known as a correlation analysis. And then we started seeing some interesting patterns here, right? Where you have a very strong correlation between split and crack seed, but I think it's pretty obvious, right? Everybody should think that way. It's just confirming, it's reassuring. There is a very strong correlation between total thickness and the palisade and the parenchyma layers based on those coefficients that you get there. You get moderate negative correlations between total seed coat thickness and split crack seeds. So what means is the more splits and cracks you have, the thinner your seed coat tends to be, regardless of the variety, whether that's slow darkening or non-darkening or whatever, right, or conventional. And the parenchyma is really, really interesting because it's showing a really, really strong correlation with that split. So we were like, okay, the parenchyma, right? There's something there that it's telling us that something is going on. We just don't know exactly. That's what happens in research, right? You do something, and then sometimes that creates more questions than what you had before, right? That's just the life of a scientist in many cases. By the way, Eduardo also do some cooking tests in collaboration with our colleague in Nebraska, Dr. Carlos Urrea. Same results that we've been saying all this time. This is just validating what we know. Slow darkening pintos cook faster, right? And again, thinking they cook faster because they absorb water quicker because of that thinner seed coat. You see how all that kind of thinking process goes, right? So what do we have learned so far about all this? There are no major differences in seed coat thickness between these two groups, right? As groups, we don't see major differences, right? Seems to be that that thickness is more variety specific. It depends on which variety you're looking at, right? And it doesn't have to be correlated necessarily with the SD trait, with the slow darkening trait, right? You have slow darkenings that had seed coats that were as good as many of the conventional pintos. But looks like the total thickness and the parenchyma layer really have to be correlated with the amount of splits and cracks that we see. So this is where we're trying to dive in more into this. When we look at those images, we have looked at many, right? We always see this kind of empty pockets, empty areas, and that's just air. And it makes me wonder, you know, from a mechanistic point of view, when you apply pressure to those air pockets, that seed coat is just gonna crack easily, right? Compared to, it's just physics, right? So, but again, we need to keep diving into that. So there's more work to do, but I think it was really exciting for me to show you the process that we've been trying to do, trying to understand and solve this problem better. We're gonna do more samples. We only have 2 years of data. We're gonna focus more on that parenchyma layer, as I was telling you about. Look at correlations with seed moisture. Maybe that's the major driver of this, as we all have said many years. Maybe we start recommending varieties with thicker seed coats or thicker parenchyma that are gonna be less prone to this damage, right? I don't know, we'll see. Doing chemical analysis, doing structural analysis. I have someone from the ag engineering department interested in doing some, applying some pressure to the seed and measuring how much force is needed to actually break that seed coat and things like that. So it's gonna be really, really interesting. So those are the air pockets I'm talking about, right? When you look at some varieties, they tend to have those empty spaces in those images, versus some others are completely full. You get the seed coat and you get the cotyledon tissue right there, right? Again, I don't have all the answers, but I want to show you how we go through this process and we get into these rabbit holes trying to figure out all these things, right? Okay, finally, I want to quickly mention our progress on white mold research, and a lot of this research is actually supported by the National Sclerotinia Initiative, which is a USDA project. We actually have the annual meeting next week, Wednesday and Thursday, right? So we all get together, different crops. This is a fungus that infects 400 plant species. 400. I'm not aware of any other fungus that can infect 400. That's how promiscuous it is, okay? That's how effective it is. So you get people from soybean, from canola, from sunflower, all kind of sitting together trying to figure out solutions for this, right? In our case, we've been playing with something that I presented before here, known as a magic population. It's a combination of 8 founder parents that you try to funnel through just an individual line, right? And try to gain some resistance in that way. We're also trying to do what we call genomic predictions using DNA data and artificial intelligence to tell us, out of these 1,000 lines, based on your DNA profiles, these 50 have promise of being highly resistant, right? Things like that. So we've been doing a lot of work with some of those lines that came out of the magic population, as I was telling you. Those are coded as WMM, that stands for white mold magic. And I have to report that we have 2 slow darkening pinto lines that are showing really high levels of resistance, both in the field and in the greenhouse, and high yield, which is the other important thing, right? So it's gonna be really, really interesting. You can see beryl on the top left is the susceptible, completely dead. And then you have some of these lines down here. We also have a small red that is doing pretty good. Michael Wunsch, he's been a speaker here several times from the Carrington station. He does these white mold trials at the station. He puts a tremendous amount of pressure in this material every year. I mean, he has the sprinting system. Some of you probably have seen what he does. I mean, I'm always hesitant to send him stuff because he always kills all my breeding lines, right? He came back and say, Juan, I think you got something interesting there. It's been 2 years in a row based on the field data. So we're pretty excited. Hopefully I can talk more final results on the next year about all these lines, but just wanted to give you a little bit of a heads up on that. More stuff that, again, in 30 minutes, I don't have time to tell you everything we do, but hopefully that gives you a good idea of all the things that we're doing. There's some additional research projects going on and different things, different problems. Popping beans. Has anybody heard about popping beans here before? I see 1 person raising the hand. Okay, 2. I'm gonna have to come next year and tell you a story about popping beans. It's quite interesting. We got a USDA grant, part of the Specialty Crop Research Initiative, to actually do some work on popping beans. So I'll probably tell you that next year. A little bit of a cliffhanger there, right? Which is pretty good, right? More traits that we're working on. We're trying to move the slow darkening gene into pinks, light red kidneys, and crans. It has been proving more difficult than we initially expected, but we're gonna keep trying on that. Last but not least, I wanted to mention something that happened last November. As many of you probably met Jim Kirkheide. He was a bean farmer around the Fessenden area. He passed away a few years ago in a boating fishing accident in Davis Lake. His family wanted to make a tribute to his life and his contributions, and they created this really nice Jim Kirkheide Dry Bean Breeding Fellowship. They made a significant contribution to the program, so we're extremely happy and humble about what they have done for the dry bean breeding program. And that shows how much value many of you put to our program and the things that we do, right? So just wanted to really share that with all of you here. Yeah. I think that's all I have to tell you. Hopefully I'm not above time. Are we good, MC? Yeah? My email is there. If you ever want to contact me or anything, there is all the list of people that really help us to move this train along. It's a lot of people behind me trying to do all this work, and we're really happy to be doing this and have the impact that you guys wanted for the bean industry. Thank you very much.